Okay, naturally it maintained the pure line or pure breeding because generation to generation they maintain same characters. This is possible because it's more always naturally it is self-pollination is the reproduction method. And the other side, the white and purple side is the same. So they have the same characters always. The other side also same characters. Now what he want to mix up now. He want to mix up two pure breeds, even though they are same species. They have a same characters, and they have the same characters. It's like a kind of like the dogs. So if we take a dog, some dog breeds are, let's say, German Shepherd. So if we do breeding, only the German Shepherd breeds are there. The other dogs are maybe Dachshund or let's say Rottweiler. These breeds are Rottweilers because people who keep dogs as pets, they want to have a pure breeds, and it's I think people call the breed is pure. We have the pedigree, and this dog is very expensive because not mixed up. It's a pure German Shepherd dog because they are doing always with the German Shepherd, and then we maintain the German Shepherd. But the other side, the Rottweilers, they maintain Rottweilers because the pure breeds are expensive. But they can mix up actually. Dogs breed can mix up because same species. But when you mix up, they share the characters again. They are half German Shepherd, half Rottweilers. I think it's similar here. Naturally, the pure breeding maintain because pollens cannot enter from others. It's mostly prevented because of this flower is very covered. So pollination happen inside the inside, and it's very difficult from the distance. Like in my drawing, I separated one side is one pure breed, other side is the other pure breed. So there are distance, there's a less chance cross pollination happen. But it is possible to do cross pollination manually. Mendel bring the pollen with the paintbrush and then open the petals a little bit, and then he can place this pollen onto the stigma. Then he can look what will going to happen if he mixed up. So he started his experiment to see the outcome, what he's going to mix up the pure breeds. So cross pollination is the method he used. Right now we come into the genetics, what we learn. Okay. Uh, then if we look the genes and alleles in our letters, the pure breeds, they have a uniform characters. So uniform characters mean it should be this form or it should be this form. Because there's no mix up. Like heterozygous condition is not mixed up. So they have a pure homozygous conditions. Over generation to generation, they maintain this kind of a pure characters. Okay, now let's start one by one to understand, right? Let's take one example, the flower color. Now, flower color is the phenotype. There should be gene for this one. There's a gene that make the flower color, and we learn gene is one, but there are different alleles. Alleles are the versions of gene. So if we take the purple and if we take a white, there should be allele combination for purple, there should be allele combination for white. Now, if we want to use our genetics, what we learned before, okay, the purple color plant, they have cell, they have a chromosome, and there should be allele here, let's say capital P, and this should be also capital P there. And if you take a white one, the white flowers, they also have a chromosome like this, and they have a simple P and simple P. Now, how should I decide capital P or simple P for purple or white? By deciding this, I automatically telling this is dominant and this is recessive because they are simple P, other one is a capital P. So I'm expressing that is the dominant and recessive. So we have to understand when Mendel is doing this experiment, he doesn't know anything. He doesn't know the chromosome, he doesn't know this allele, he doesn't know this capital, he doesn't know dominant, he doesn't know recessive, nothing. But for learning purpose, we mixed up now our knowledge and what Mendel did. So we can learn two different ways. One is we can learn completely what Mendel did, and after that we can interpret. Okay, Mendel got this result because of this. But it's very difficult to explain if we use completely Mendel method because we can't write anything to explain. We can only show the result at the end. But when we combine our knowledge, it is to understand what Mendel did. Let's start his first experiment then. These are the purple flowers. And this may be the white flowers. So he crossed these purple and white flowers. He did the cross pollination to see outcome of the next generation. So we can say this is the parents, and then the next generation is the offspring. He observed it's always purple. It's always purple. Now again, Mendel did one experiment, just one experiment, and he see purple color. Then Mendel understood, okay, this time purple. Maybe next time I will get white. Then again he did second experiment. Oh, this time again purple. I didn't get white. He did third experiment. This time again purple. Then he at least did not give up. He tried to go again the fourth and fifth one. Purple, purple. Maybe after 30 times or 40 times, any stupid person can understand I never get white. So Mendel ended up, okay, I will never get white, I'm always getting purple. So he concluded after maybe I don't know how many experiment he did. Maybe after 50, 60 experiment, he gave up. No, this doesn't work. I'm always getting purple, right? So this is we going to write as a first generation, right? First generation is like this F1. Now why we write F1? F is indicated for filial. Filial, yeah, filial. Filial is the meaning is again like a siblings, like offspring generation is F1 generation. So Pendel first experiment, all get purple. Let's assume he did 100 times this experiment and he never get white one. Now for all of us it's confirmed we never get white one because it's always purple. I think next step is give up the experiment and then he's doing what he wants to do. Just go to the church and his priest, he will do his job. But I think one of the strong character scientists has they never give up. Scientists never give up until they find what they wanted to see. I think the famous story, maybe you have heard about Thomas Edison said, I failed 999 times and I success only one time to discover the bulb, something like this. So he learned each time when he failed 999 times. So same thing, uh, he can easily give up now. Why should I spend time doing 100 times this plant? Stupid thing, I get always this. But then he changed a little bit. He changed. What he changed, this F1 generation, right? This is the F1 generation. He thought something to change now. Why I should change now? This F1 generation always purple, right? So I write it here purple. This is white, all purple. So then he took F1 generation, this one, and this one. It's again purple and purple, right? So we can say this is purple, this is a purple. So he did one experiment, he got again purple. He did another experiment, it's again purple. Now it's again chance to give up because nothing is working, it's always purple. But he kept on continuing. I don't know actually the exact numbers, but when I realized, like, see this experiment, like he did hundred of experiment. He was lucky in one experiment. He observed white. He observed white again. Then it's motivate him to do and do more and more experiment. So he did tremendous amount of experiment. So he did one set of experiment, ended up like this. Uh, 299 white, no, just so I just some kind of assumption numbers, not his real numbers. So he has seen white flowers 101, and the purple 308. And he did again another set of experiment. He did again one experiment 99, and then 300 and 300. And he did another set of experiment 299, 100. So I'm taking some example like that we can learn something, not these real numbers, right? Then it 300, 301 and 107. Okay, let's see four experiments, set of experiment. Can you observe something from these numbers?
Yeah,
What is
raised
it?
one,
Hmm?
raised one ratio. Yes, sir.
Yeah, he did the same thing. There's a pattern. There is something similar in each experiment. But I get 3 to 1 ratio, close to 3 to 1 ratio. Now that's where the scientists try to find this kind of a miracle things by observing very carefully and analyzing the data. She got 3 to 1 now. So, so this is F2 generation. The F2 generation, filial 2 generation result, 3 to 1. Now, you can assume if he doesn't know about genetic, he doesn't know about the chromosome, he doesn't know about the DNA, he doesn't know about this kind of a thing. Now, can you assume that he has only this result? This is his observation. Now, imagine that he came up with the like all these theories by looking at the result. So this was his result. So how can he make some kind of a theories and some kind of a laws or rules from this result? His capabilities. So he came up with the ideas. He had only this result, only just all in F1, F2, 3 to 1 ratio. I think we can uh, add something how this is happening, but we directly go to our known genetics to explain these rules, explain this result. And if he wants to explain, he might say, well, there is a purple flower in the first generation, something is coming from purple flower, and it's always giving purple, white flower cannot make it. But something is also coming from white flower, that's why this side has at the end the purple, this side is purple, and this side is white. So he could explain like this, now something is coming from purple, something is also coming from white one, but the purple one is strong, it is making the color. In F2 generation, there is a chance that the white color also can appear because white one is also there. It is also making white, but purple one is stronger, it make more. So that was his kind of explanation. So some factors. So he said some heredity factors coming from purple one. One heredity factor is coming from white one, and they combine together. But purple one is giving. When I'm doing the F2 generation, then again they separate these factors, and the factors can again join together. So purple one is stronger, it gives more. White one can also join and it can give a white color. So it's try to explain like this, go on, when we can go on like this, but at the end, our genetics, what we know, we can easily explain because we learn and we know these genetics, chromosomes, all these things. So coming back, it is like this.
So,
Yeah.
second time when he did the self-pollination and cross-pollination, sir.
He did mostly the cross pollination. So
Okay.
he select, let's say he had 400 flowers, then he cross pollinate 400 flowers. I think it was so much of work. At the end, there was so much of discrimination from the other people because some people think this guy is a crazy, why he's doing with the flowers this kind of a thing, and what he want to do these things. So people are against actually uh, on his work, what he want to show. And if I read his history, his document destroyed by other people, somehow he was lucky enough to bring into the scientific community, and not that easy to convince them because being a priest, nobody accept that he's a scientist, but somehow it worked. Yeah, it mostly did the cross pollination. So in real experiment, you can understand they had to take the plants, 400 plants, and then he did all in that set cross pollination. Then he observed this plant, and also you can understand in real situations some plants are dying, like some plants are, it's not all plants are surviving, some dying. We just say like easily when we are learning genetics, okay, it's a 3-to-1 ratio, but this 3-to-1 ratio is, there are probabilities like a chances. So if something happened, we have to talk about the chances as well, probabilities. Finally, it is if we take large numbers, one of the success of his experiment, he used large sample size, like this much of sample size, 1,500, like 600 times large sample he used to get these ratios. So more samples, the ratios are getting very closer. Okay, there's another thing now we're coming up. The breeding experiment, we add a little thing to our standard way. We call it one. There's a flower color, right? It's a one character, and then one character come in different ways, purple and white. So when we cross purple and white, it's a hybrid now because purple plant and white plant. So one character hybrid. This one character hybrid, one character is mono hybrid. So we can consider now in a standard way, the first set of experiment is monohybrid cross. Monohybrid cross. So in this case, we need to learn standard things about how to write exactly the correct genetic diagrams like things, understanding this monohybrid cross. Yes, another important thing, uh, many genetic diagrams, uh, they have a different method to use, but we try to use one type of genetic diagrams. Okay, we take the color then, purple. So we here it's not real genetic diagrams, but this is the experiment he did. So we try to make the genetic diagram for this one. First, parents. So we have to write the parents' phenotype. Example, purple and white. Purple and white. And then we have to write parents' genotype. Second one, parents' genotype. Purple one was the dominant one in his experiment. So like this, and I use simple P for the white one. Then in meiosis, the gametes are formed. That's the ovum and the pollen. When the gametes form, according to our knowledge, they separate, right? So separate mean a capital P, simple P. And then what will happen uh, if this is a gamete, this is a gamete, this is a gamete, this is a gamete. Now this gamete can fuse with this gamete. If it is the case, this is the outcome. This one can also fuse with this one. So same with this. So whatever the outcome, it's always this. So this is F1 generation, and this is the phenotype. It's blue, purple, right? All purple. Now what we did, we use our modern knowledge to explain the Mendel's genetics, and we get all purple. Now next time, F2 generation, this one I should, F2 generation, I should get this. Capital P, simple P going to cross with capital P, simple P. So if there are parents here, they are purple now. And the gametes, P, simple P, capital P, simple P. Now in this situation, we use a little bit of standard method, that is called Punnett square. So male and female. Now I can place the gamete here. Uh, let's assume this is a male flower, this is a female flower. Capital P, simple P. Capital P, simple P. Combination is capital P, simple P, simple P, simple P. Okay, the Punnett square is some easy way of showing, otherwise nothing special. Just give me 1 second now. Right, the Punnett square is a graphical representation of the data. The person name is Punnett, and he proposed you can use this kind of a table. It's easy that you can get your result, and which help you to understand easily, right? So this is capital P is a purple, capital P simple P is a purple, and this is a purple, and this one is white. So ratio is 3 to 1. So we get 3 to 1 ratios, 3 purple, 1 white. I don't have a space, I write here. Genotype ratio, capital P is 1. This we have a 2, 1. Phenotype ratio 3 to 1. 3 purple, 1 white. Right. So this is the end result. Okay, I think we make it shorter. So he did many experiment, not only the flower color. He can take the seeds, he can take the pods, the height of the plants. So he did many experiment, and finally he observed this is the result. So you get always this kind of a result. So at the end, what is the conclusion? This is the allele. This allele separate, and then they form gametes. So he make his first law. of segregation. So, law of segregations, what he proposed, the allele are separated and move into each gametes. And get into each gametes. When gamete formation. When forming the gametes, alleles are separated and going to each gamete. So it is this one. So yes, this one. Here one gamete, here one gamete, and here one gamete, and here one gamete. So alleles are separated and get into each gametes. Alleles are separated and get into each gametes. So this is the first law of Mendel, law of segregation. Alleles separate and get into each gametes when gametes are forming. So based on this is the point. He formulated his first law. Alleles separate and get into each gametes when gametes are forming. Okay, after analyzing everything at the moment with our modern knowledge of genetics, we accept this is a first law of Mendel, law of segregation. Sometimes you will see this law is describing in different way, but as for the resource book, we keep like this: alleles separate and get into each gametes when gametes are forming. That's a law of segregation. Right. Yes. So, do you have any questions? Everything clear?
Clear, sir.
I will upload everything to the site and I send you the password, right? Password. Okay, I can text the password after that. So you can go to the site and genetics. Then you see where you have to go. The node, this node, and the other node. If I mean, it's a mainly resource book. Nothing much. Sometimes I add little thing, but otherwise 99% is resource book. One of the thing, some student they are going to different teachers. They learn from Campbell and this thing. One of the student told me, sir, I went to that class. He's always we learn everything from Campbell book. At the end, so it is good for knowledge. But the exam, I got a B because I can't remember that much and I can't write what exactly in the resource book. That like that. I mean, we accept resource book is the only material we have to use, but for understanding purpose, we may use little things. But everything at the end resource book, right? Okay, then we'll continue law of second law in the next class. If you have any question, you can text me, right?
Okay, sir.
Okay, Usman, then take care. See you.
Thank you, sir.
Okay, bye.